Hello to everyone. Today's topic for discussion is digestion and absorption of proteins. Introduction. What are the proteins? Proteins are the polymers of L-alpha amino acids linked by peptide bond. Proteins are the primary constituents of the body. A regular and adequate supply of protein in the diet is essential for the cellular integrity and functions of the body. On an average, adult man requires 70 to 100 gram of proteins per day in the diet. And these dietary proteins are the primary sources of amino acids. What is the function of amino acid? The main role of amino acid is in the synthesis of structural and functional protein. Carbohydrate and fats can be stored in our body, but proteins are not stored in the body. Proteins are made up of two types of amino acids, non-essential amino acids and essential amino acids. Non-essential amino acids are either derived from the diet or synthesized in the body, whereas essential amino acids are not synthesized in the body and should be supplied from the diet. Even if one essential amino acid is deficient in the diet, protein synthesis cannot take place. Digestion of protein. What is digestion? Digestion is the disintegration of complex nutrients into simple, soluble and assimilable form. Proteins are large polymers of alpha amino acids. So, these proteins cannot be absorbed easily. The dietary proteins are denatured on cooking and hydrolyzed to amino acids by proteolytic enzymes. So, after that they can be easily digested. All the enzymes which causes hydrolysis of protein belong to hydrolase group of enzyme classification. These enzymes are called proteolytic enzymes. They are secreted as inactive zymogens which are converted to their active form in the intestinal lumen. This would prevent the autodigestion of secretory acini. You can see here the structure of protein, which is made up of number of amino acids linked by peptide bonds, and these peptide bonds undergo breakdown by the action of proteolytic enzymes, which are also called as proteases or peptidases. And after the action of proteases, peptidases or proteolytic enzymes, individual amino acids are formed. These amino acids can be absorbed easily. What are the characteristic features of proteolytic enzymes? Proteolytic enzymes belong to hydrolase group of enzyme classification. These are of two types, endopeptidases and exopeptidases. What do you mean by endopeptidases? Endopeptidases are the proteolytic enzymes which act on the peptide bonds inside the protein molecule. Examples are pepsin, trypsin, chymotrypsin, elastase. Second type is exopeptidases. What is exopeptidases? They act at the peptide bond only at the end region of the chain. Two types are there again of this exopeptidases, carboxypeptidases and aminopeptidases. Proteolytic enzymes are secreted in inactive form. This inactive form is also called proenzyme or zymogen. And when this zymogen reach the site of action, they are activated. This is general overview of digestion of protein. There is no digestion of protein in mouth because there are no proteolytic enzymes in salivary juice. Digestion of protein begins in stomach. Then it continues in intestine by the action of pancreatic juice and intestinal juice. In intestine, after digestion, there is absorption of protein also. Enzymes involved in digestion of proteins. In mouth, there are no enzymes. In stomach, there are two enzymes, pepsin and renin. Pancreas is responsible for the secretion of number of enzymes, and these enzymes are trypsin, chymotrypsin, elastase, carboxypeptidase A and carboxypeptidase B. Intestine is also responsible for the secretion of two enzymes, aminopeptidase and dipeptidase. Digestion in mouth. As you know, there are no proteolytic enzymes in the saliva, so no digestion of proteins in the mouth. But in mouth there is secretion of saliva. And what is the function of saliva? It lubricates the food, which helps in making the food soluble for the action of proteolytic enzymes. After mastication, food reaches stomach where it meets the gastric juice. Digestion in stomach. Digestion of protein basically starts in stomach. When protein enters the stomach, it stimulates the secretion of hormone gastrin from the gastric mucosal cell. And this gastrin is responsible for the release of gastric juice, which contains hydrochloric acid, pepsinogen and renin. What is the pH of gastric juice? It is 1.5 to 2.5. Stomach is responsible for the secretion of strong acid, that is HCl, and an enzyme called pepsinogen. Pepsinogen is a zymogen, or it is also called proenzyme. It is a inactive enzyme. What is the function of HCl? HCl is responsible for denaturation of proteins and making their internal bonds more accessible to subsequent hydrolysis. It is also responsible for providing decreased pH. This decreased pH is responsible for activation of the pepsinogen to pepsin. HCl also kills some bacteria, so it is having bactericidal action. HCl helps in the absorption of vitamin B12. And the inactive enzyme pepsinogen, which is secreted by chief cells of the gastric mucosa, is activated to pepsin by the action of HCl. When pepsin is formed, this pepsin is responsible for conversion of inactive pepsinogen to pepsin. This is autocatalysis. What is the action of pepsin? Pepsin acts on peptide bonds formed by carboxyl group of phenylalanine, tyrosine, tryptophan and methionine. Pepsin is endopeptidase enzyme. It acts on internal peptide bond. It converts protein into proteases and peptones. Pepsin cannot act on proteins like keratins, mucoids and mucoproteins. There is another enzyme secreted by the stomach, and this enzyme is called renin. Renin is also called chymosin. The activity of renin is seen in infants only. Renin is absent in adult stomach. Renin is responsible for curdling of milk. It converts casein of milk into paracasein. It combines with calcium to form paracasinate. Renin secreted in the stomach should be differentiated from renin secreted by the kidney. Renin secreted by the kidney is a proteolytic enzyme, and it is involved in the activation of angiotensinogen to angiotensin, which is a hypertensive agent. Digestion of proteins in the intestine by pancreatic enzymes. As the acidic content pass into the duodenum, the low pH triggers the secretion of the hormone secretin. This secretin is responsible for stimulation of the pancreas to secrete bicarbonate, and bicarbonate neutralizes the HCl and raises the pH from 1.5 to 2.5 to 7. There is a release of another hormone called cholecystokinin, which stimulates secretion of pancreatic enzymes. And these enzymes are of two types, endopeptidase and exopeptidase. Endopeptidase are trypsin, chymotrypsin, elastase, which are secreted from pancreas. Whereas exopeptidase, which is secreted from the pancreas, is carboxypeptidase. Aminopeptidase is secreted from the intestinal mucosal cell. Endopeptidase are secreted from the pancreas, and they are responsible for breaking of internal peptide bonds and results into formation of smaller peptides. What are the endopeptidases secreted from the pancreas? These are trypsin. Trypsin is secreted in the form of inactive zymogen, trypsinogen. Chymotrypsin, it is secreted in the form of chymotrypsinogen. Elastase, it is secreted in the form of proelastase. Exopeptidase secreted from the pancreas is responsible for cleavage of one amino acid at a time from either C or N terminal of the polypeptide chain. There is only one exopeptidase secreted from the pancreas, and this exopeptidase is carboxypeptidase. It is secreted in the form of procarboxypeptidase zymogen. Now we'll see the action of trypsin enzyme. Trypsin enzyme is secreted in the inactive zymogen form trypsinogen from pancreas. Trypsinogen is activated by enterokinase, which is secreted by the intestine, or it can be activated by trypsin itself by autocatalysis. Trypsin is responsible for cleaving of peptide bonds contributed by lysine and arginine, that is, peptide bonds contributed by basic amino acids are degraded by trypsin. Trypsin also acts on zymogen forms of other pancreatic enzymes and activates them. For example, chymotrypsinogen is converted to chymotrypsin, proelastase is converted to elastase, and procarboxypeptidase is converted to carboxypeptidase by the action of trypsin enzyme. Trypsin enzyme has weak action on casein. Next we will see chymotrypsin. Chymotrypsin is secreted in the inactive zymogen form, that is chymotrypsinogen, which is activated to active enzyme chymotrypsin by the action of trypsin enzyme. And what is the action of chymotrypsin? Chymotrypsin hydrolyzes the peptide bond formed by carboxyl group of aromatic amino acids. What are the aromatic amino acids? Phenylalanine, tyrosine, tryptophan. Elastase. Elastase is secreted in the inactive form proelastase from the pancreas. Proelastase is converted to elastase by the action of trypsin enzyme. And what is the action of elastase enzyme? It acts on peptide bond formed by the amino acids like glycine, alanine and serine. Now we'll see carboxypeptidase. Carboxypeptidase is exopeptidase. It is secreted from the pancreas. It is a zinc containing enzyme. It is secreted in the inactive zymogen form procarboxypeptidase. This procarboxypeptidase is converted to active form, that is carboxypeptidase, by the action of trypsin enzyme. Carboxypeptidase cannot act on dipeptides. Carboxypeptidase is of two types, carboxypeptidase A and B. Carboxypeptidase A acts on the carboxy terminal peptide bond connected by tyrosine, phenylalanine or tryptophan, whereas carboxypeptidase B acts on the carboxy terminal peptide bond connected by arginine and lysine. You can see here overview of digestion of protein. You can see in this picture how inactive zymogen is converted to its active enzyme form, how pepsinogen is converted to active pepsin by HCl, how trypsinogen is converted to its active form trypsin by the action of enterokinase enzyme. You can also see here how various proenzymes like chymotrypsinogen, proelastase and procarboxypeptidase are converted to its active form, that is chymotrypsin, elastase and carboxypeptidase respectively by the action of trypsin enzyme. You can see the peptide bond on which these enzymes act and cause its breakdown in this figure in detail. Digestion of proteins in the intestine by intestinal enzymes. The digestion products of hydrolysis of pepsin, trypsin, chymotrypsin, elastase and carboxypeptidase are completed by intestinal peptidases secreted by mucosa of small intestine. And what are the intestinal enzymes? These are aminopeptidases and dipeptidases. Aminopeptidases and dipeptidases both are present on the luminal surface of intestinal epithelial cells. Aminopeptidase acts on N-terminal peptide bonds and release free amino acids, whereas dipeptidase acts on dipeptides and releases free amino acids. After digestion, now we'll see absorption of amino acids. Amino acids are absorbed from the ileum and distal jejunum. Oligopeptides like di and tripeptides are absorbed from duodenum and proximal jejunum. Absorption of amino acid is an energy requiring process and energy is given in the form of ATP. Absorption of most of the amino acids take place by sodium dependent active transport system. For example, sodium amino acid co-transport system. This transport system is responsible for transport of both sodium and amino acids. There are 5 different carriers for amino acid transports: small neutral amino acid transport, large neutral amino acid transport, basic amino acid transport, acidic amino acid transport and amino acids and glycine transport. Now we'll see the sodium amino acid co-transport system. This transport system is responsible for simultaneous transport of both sodium and amino acids inside the intestinal cell from intestinal lumen. For this transport, energy is required, and this energy is given by ATP. You can see here 5 transport system. A small neutral amino acid transport system is responsible for transport of alanine, serine and threonine, whereas large neutral amino acid transport system is responsible for transport of isoleucine, leucine, valine, tyrosine, tryptophan and phenylalanine. Basic amino acid transport systems are responsible for transport of arginine and lysine. Along with that, ornithine and cysteine are also transported. Acidic amino acids are transported by acidic amino acid transport system, and these are glutamic acid and aspartic acid. Amino acid and glycine transport system is responsible for transport of proline, hydroxyproline and glycine. Absorption and transport of amino acids in intestine, liver and kidneys across the mucosa occurs through gamma glutamyl cycle, which is also called Mister cycle. This cycle occurs in intestine, brain and kidney, and glutathione is required for this cycle. This glutathione is a tripeptide made up of glutamic acid, glycine and cysteine. At brush border, glutathione combines with amino acids in the presence of enzyme known as gamma glutamyl transferase enzyme and forms cysteine glycine and glutamyl amino acid complex. At the end of the reaction, glutathione is regenerated and one amino acid is absorbed across the cell membrane. The transport of one molecule of amino acid and regeneration of GSH, that is glutathione, requires 3 molecules of ATP. Now we will see this reaction in detail. Amino acid present outside of the cell membrane is transported across the cell membrane with the help of glutathione and gamma glutamyl transferase enzyme. Glutathione combines with L amino acid and forms 2 complexes, cysteine glycine complex and glutamyl amino acid complex. Glutamyl amino acid complex undergoes breakdown and releases amino acid and forms oxoproline. This oxoproline utilizes ATP and get converted into glutamate. Glutamate again combine with cysteine and form glutaminyl cysteine residue. This reaction also requires ATP. Glutaminyl cysteine residue later on combine with glycine and form glutathione. So glutathione is regenerated and amino acid is internalized. Absorption of intact proteins and polypeptide. For a short period, immediately after the birth, small intestine of infants can absorb intact proteins and polypeptide. This process may occur by endocytosis or pinocytosis. The direct absorption of intact protein is very important for the infant for the transfer of maternal immunoglobulin, that is gamma globulin, to get immunity. Intact proteins are not absorbed by adult intestine. However, in some adults, this process may occur and responsible for antibody formation and food allergy. Clinical significance associated with digestion and absorption of protein is acute pancreatitis. What is pancreatitis? Pancreatitis is nothing but inflammation of the pancreas. It may occur due to premature activation of the trypsinogen inside the pancreas. Trypsinogen may get converted into trypsin, and this trypsin causes breakdown of peptide bonds present in the protein of pancreas. That leads to destruction of the pancreas, and this process is called autodigestion of the pancreas. This is a life-threatening severe condition, and if not treated, it may become fatal. Defects in the intestinal amino acid transport system lead to development of 2 inborn errors, cystinuria and Hartnupp's disease. In cystinuria, there is one common transporter for cysteine, ornithine, arginine and lysine. This transport system is present in gut and renal tubules. Because of the defect of this transport system, there is loss of these amino acids in feces and urine, and this condition is called cystinuria. In Hartnupp's disease, there is a deficiency of transporter for tryptophan and neutral amino acid. Because of the deficiency of transporter system, there is no absorption of tryptophan. So tryptophan is excreted in the feces and lead to its deficiency. Tryptophan deficiency produce neurological and skin manifestation just like pellagra syndrome.